It's a great pleasure to remotely present this lecture, and I had a great time visiting IIT Montreal last year. So the topic for today's lecture is the same as last year. I modified the content towards the end of this lecture. During the lecture, if students have a question, please use a microphone so I can hear clearly your question. What is a spatial hearing? So spatial hearing is the sensory ability allow us to know where things are through sounds. This ability is extremely important for survival of a species, especially for animals, in terms of finding food and avoiding threats. For humans, spatial hearing is also very important, because in a natural listening environment, we often not just listen to sounds coming from one source. We're often surrounded by many speakers. Sometimes in a very loud environment, there's ambient background noise. In spatial hearing, the ability to know where things are help us to direct our attention to important events in a crowded scene. And this area study was coined by E.C. Cherry almost half a century ago, called the cocktail party phenomenon or effects, talking about importance of auditory spatial attention for auditory scene analysis. Spatial hearing is also important for detecting event coming from a location outside our eyesight. So what you see in this painting is describe ancient wisdom by a Chinese philosopher Zhuangzi. What shows here is a mantis stalked a cicada. So although he's thinking he's within striking distance to his food, he's unaware of the yellow bird behind. So we use this idiom to describe situation where we only focus on rewards or immediately in front of us, but somehow forgot about the consequences or danger surrounding or behind us. So for today, we have 3 areas of topics to cover. First, we'll talk about the computational goal of spatial hearing, and we'll learn acoustic cues for sensory localization. This regarding the physics of spatial hearing. Then we will talk a few selected topics in anatomy and physiology of auditory spatial processing regarding neurophysiology. The first 3 area of materials are similar to what I covered last year. The differences will be the last quarter. I will talk about some ongoing research in my laboratory. So if I would ask you a simple question, where's the puppy? And I think your answer will be very straightforward. You will say, it's right there in front of us. Yes. So the reason we can easily identify location of a visual target is because our visual system is established, and from the very beginning or first stage, the spatial sensitivity for future analysis. The so-called receptive field structure, I think has been covered in early lectures. And essentially, the photoreceptors inside our retina has already have this spatial sensitivity or selectivity, and different receptors will have sensitivity to cover different regions of space. This spatial sensitivity and selectivity is passed on to the downstream process up to many stages of visual cortex. The spatial sensitivity is very important for visual processing, and especially for visual object recognition. If I would remove this spatial information, we will no longer know where is a visual object. So in this case, we do not see clearly where individual object is because of occlusion. So the question is whether or not occlusion also a difficult situation for audition. To realize this, let's hear a sound clip, and let's try to envision what is the situation the sound actually try to convey in this simple listening exercise. Oh, go too fast. So just 4 seconds of a sound, we actually can envision is a pretty grooming situation. Hopefully you will hear there's a dog barking, there's a thunderstorm, and hopefully you can also hear there's a distant sound on a rainy day. So depends on speaker setup in your lecture hall. There's no spatial information is provided in this sensory input to your ears. However, we can vividly reconstruct this visual image in our head or in our mind. There's so many different things happening. So this is the beauty of auditory scene analysis. Auditory scene analysis, or the ability to detect and discriminate different auditory object, is not necessarily depend on the spatial information of object. So this topic is beyond the scope of the lecture, and for students interested in this area of research, here are two very useful textbooks for references. So what is receptive field structure for audition? This topic will be covered in next lecture by Dr. Xiaoxing Wang. Here briefly, inside our cochlea, which is our sensory receptor for audition, for hearing, but because the mechanical property of basal membrane, different parts of basal membrane has different frequency selectivity, as shown on this line. So the basal end of basal membrane will respond to high frequency of a sound, and the apical end of basal membrane will respond to the low frequency of a sound. This is simply due to the mechanical resonance properties of this rubber band, essentially. This frequency selectivity will be passed on, actually stage by stage, through the ascending auditory pathway, and even in auditory primary auditory cortex, we can see that different area of auditory cortex will respond to different frequency of a sound. This structure is called tonotopy, meaning that the spatial range, the frequency selectivity neurons are spatially arranged, so that by looking at different region of brain or different region of brain inside the nucleus, we know actually which part of the sound are encoded. So that being said, the computational challenge of auditory perception is not simply due to the fact that there are many sound sources in the space. Instead it's because sound itself does not have any spatial dimensions, unlike many visual objects, and is required auditory system to reconstruct an entire auditory scene using only 2 channels of sound information. So here are the 2 channels referring to, we have left and right ears. Our sound information coming from different sources essentially are mixed, is in a melting pot, and the mixed information sent to both ears. Auditory system has to untangle essentially all the sound information in multiple dimensions and recover what is the information coming from different sound sources. So how does auditory accomplish such computational feat? And actually, this problem has been realized for over 100 years, and it's through studies of psychoacoustics, which link the physical world with our perceived world. And a lot of study has been conducted, realizing what are the processes are needed for successfully constructing a perceived internal representation of external world. And through advances in modern brain technology, we know more, and especially through the technology of electrophysiology and computational modeling, we know more about how this internal process in the brain, essentially the neural processing, allow us to realize location of a sound and knowing how many sounds in the space and who's talking from where, essentially. So for next segment, we'll be focusing on the physical world, essentially the auditory localization cues to begin with, and in terms of auditory spatial hearing and spatial computation. So if we would divide the space around us in terms of left and right, front and back, and up and down, actually different cues are used for identify a different spatial dimension of a sound. In azimuth, which is the ability to tell sound coming from left or right, we use interaural difference cues in terms of timing and the level differences between 2-year signal at 2 years. And for elevation, in other words, the ability to tell sound coming from above or below, we rely on Monaural spatial cues. And for distance, near or far, we rely on relative loudness and reflection information coming from the surfaces surrounding a sound source. Next, I will focus on the two sets of cues. One is monaural, the other is binaural cues. So monaural cues, as you see at panel A, is simply due to the fact that our outer ear, if we would touch your ear right now, you realize the ear, outer ear shaped like a funnel, right? And we all know the funnel can collect the sound, amplify sound energy. But if you look at the ears of your neighboring student, you'll see that the outer ear actually is not symmetrically shaped in terms of the structure of below and above the ear canal. Due to this asymmetry, when sound waves reaching at the ear canals, it will create reflections around by the rim of our outer ear. Due to the asymmetry, the sound coming from different elevation angles will create a different resonance will create different reinforcement and cancellations between direct and reflection waves. So this reinforcement and cancellation will create those bumps and troughs in this monaural filtering profile. So this is actually the filtering of the monaural process has a word for it called a head-related transfer function. Is widely used in audio engineering and in also virtual three-dimensional spatial hearing. So it's a study have shown that the location of the dip were called spectral notches. Actually is correlated with the elevation angle of a sound source. For sound coming from below, the notches happens at the lower frequencies. As the sound source move up, these notches will move to higher frequencies, simply due to the asymmetry shape of the outer ear, or pinna, as I mentioned earlier. And these monaural or spectral notch cues provide information about elevation. So panel B and C shows the two binaural cues. One is called interaural time difference, or ITD. The other is called interaural level difference, or ILD. They are very simple concepts. So simply speaking, when sound coming from one side of the head, and for low frequency sound, it will create a different travel length, reaching at left and right ear. For example, in this case, sound coming from the right side. Therefore, sound wave will reach the left ear at a longer distance, therefore, rather a later time. And if we would measure the sound waves at ear canal, we'll realize the sound coming from the further ear will delay in time. So this level time delay create a cue for the auditory system to compute the lateral direction of sound source if the sound is a low frequency has low frequency. Because at high frequencies, the auditory neurons cannot follow cycle by cycle this level time information. Therefore, the interaural time cues, even though exist in physical dimension, cannot be used by the nervous system. So ILD simply means is in level differences in signals reaching at 2 years. In this case, the far ear, which is on the left, receive the sound after sound has passed its head. So due to the Hashira effect, which is obstacle in the path of sound traveling, will attenuate the sound energy. So if we would measure the sound waves at the ear canal, we realize at the far ear, not only the sound arrive later time, but also is attenuated in amplitude. ILD is only significant cue for high frequency sound. Is it because at low frequencies, sound simply can bend over or pass the head without losing much of energy. So because of this, ILD is useful only for localizing high frequency sound. So decades of research has realized the three cues, ITD, ILD, and Monaural spectral notch cues, are first encoded by neurons in the brainstem of auditory system, especially superior olive for encoding binaural cues, ITD and ILD, and the cochlear nucleus, especially the dorsal part of the cochlear nucleus, are responsible for encoding the spectral cues. For time, concerning the time of this lecture, next we'll focus on the neurocoding mechanisms for ITD and ILD. So the mechanism of ITD computation actually has been speculated more than half century ago. It was a famous model called the Jeffress model. In late 40s, Jeffress proposed perhaps the reason we can utilize the interval time difference or interval phase difference is because the external delay differences, ITD, can be compensated by the internal travel time of the signal from the ear to a neuron or to a central processor. And if these two external time differences and internal time differences are somehow just equal to each other but reverse in sign, then the signal arriving at the individual cells will arrive at the same time. So if the cell has a capacity to detect the coincidence of a two-size input, then the cell perhaps can fire, maximally to signal arriving of some information. So this elegant model actually was proposed before we know anything about physiology. And physiology confirmed this model in 1960s in mammalian species and demonstrated elegance of a thought experiment. So in this diagram is taken from available textbook neuroscience and about this Jeffress model. So in this case, sound coming from the left hemisphere will reach the left ear earlier than sound reaching to the right ear. But inside our brain, the signal will travel longer time from left ear to a central structure called later identified called a medial superior olive, and different MSO neurons will have a different arrangement of an internal delay line, so that different cells will encode different external ITDs. So the captions or legends of this graph demonstrate this multiple steps process, and I think it explains the mechanism really well. So for students interested in this mechanism can read later about this model. So as I mentioned, physiology identified these confirming mechanisms later on in mammalian species. What you see here is a slice taken from the gerbil of MSO. Inside this MSO, there are many neurons arranged in parallel, and each neuron, they have very unique structure, a simple but unique structure. They have this bipolar cell shape. They have a giant cell body, but then have two dendrites, one as arranged laterally. And the amazing feature about MSO cell is the two dendrites receive inputs coming from different ears. And there are specialized ion channels along the dendrites and inside across the cell membrane of the cell body, enable very fast membrane time constant. Fast, I mean, in the sub-millisecond time scale, to allow this computation of rather times of two inputs coming from different ears. And then in recent decades, the also study indicate not only this input carry excitatory information coming from left and right ear, also the MSO cell receive glutaminergic inhibitory input coming from the cochlear nucleus. And inhibitory input are believed to has ability to modulate further